Hello students, today we will be starting with the new unit, the second unit. The chapter is Principle of Inheritance and Variation. So here we will be studying about the Mendel's laws, chromosomal disorders, blood groups, and today we will be studying about the terminologies. And let us see who is Mendel. So one by one let us study, and this lesson will be having many sessions. It's the lengthiest chapter. Now look at this photo, children. Can you see there that child's eye color is blue, like her parents' eye color itself. So now see the hair of the child. It is brown, whereas parents' hair color is black. So what is the mean? Why there is such differences? So for these questions you will be getting answer from this genetics. So genetics is a branch of biology that studies with the inheritance and variations and characters among related organisms. So largely in the evolutionary aspects. So inheritance is a basis of hereditary passing from one offspring to another. So the offspring differs from its parents. Human beings have been using their knowledge of genetics since prehistoric times. So our ancestors, they knew that sexual reproduction was one of the causes of variation. Based on this principle, we practice domestication and selective breeding of animals and plants. For example, now the cow which is found in Punjab is attained through artificial selection and domestication of ancestral wild cows. However, our ancestors, they had very little knowledge about the scientific basis of genetics. So it was only during the 19th century there were some advances made in the field of genetics by Gregor John Mendel. So Mendel, he was an Augustine priest and scientist who gained fame in spreading the science of genetics. He proposed many laws of inheritance by conducting extensive research and hybridization experiments on garden peas, by P. setium for about 7 years. So hybridization is the process of crossing two individuals differing in at least one character. So resulting in the hybrid individuals. The reason for Mendel choosing common pea plant for his research was that the plant had various contrasting characters like length of the plant or color of the pod and shape of the seed, so that were visible to the naked eye. So moreover, self-pollination as well as cross-pollination was possible in these plants, and crossing between these plants controlled. In addition, these plants grow quickly and they produce large number of offsprings. So Mendel, for the first time, what he had applied some statistical analysis and mathematical logic for this biology. So he chose 14 ways of true breeding, 3 lines similar in all aspects except for one character, to conduct cross pollination or artificial pollination experiments. So here a true breeding line, it exhibits a stable inheritance and expression for many generations due to continuous self pollination. Round or wrinkled seeds, yellow or green seeds, green or yellow pods, or tall or dwarf plants. These were some of the contrasting traits which was chosen by Mendel. And the use of true breeding lines helped Mendel for his basic framework of rules relating to inheritance, which were later expanded as scientists discovered new information pertaining to genetics. So also the data collected by Mendel was quite credible as he had used large sampling size for his experiments. So moreover, when he conducted experiments on several generations of test plants, it helped him to confirm his influence and prove his rules were not confirmed ideas but general rules of inheritance in his own living organisms. However, the world had to wait for the end of the 20th century to understand the significance of Mendel's discoveries. So because of his valuable research in the field of genetics, Mendel came to be known as father of genetics. And rediscovery of his laws laid to the foundation of the modern science of genetics. Now let us study the technologies while studying genetics. So firstly let us study the term a gene or a factor. It is a functional unit of hereditary material, that is DNA. So in other words, it is a unit of DNA that is responsible for the appearance and inheritance of the character. The alternative form of the gene located at a specific position on a specific chromosome is known as the allele. It governs the trait of the individual and is generally represented by a letter. The trait can be defined as a distinct variant of an organism's phenotypic character which may be inherited or environmentally determined. So example, see the color of the petals is a character or an attribute, while the colors pink, violet and white, these are the traits. The genetic makeup of an organism is known as genotype, while the appearance of the organism is denoted by the term phenotype. Every organism possesses two alleles for each character. If these two alleles for the particular character are identical, the gene is said to be homozygous or pure, and the individual is called a homozygote. Every organism possesses only two contrasting characters of different alleles in a pair. The gene is called heterozygous, and the individual is called the heterozygote. So in this heterozygote, there is only one of the contrasting genes is able to express itself, while the other gene it remains hidden. So the gene which expresses itself in the F1 hybrid is called the dominant gene, while the gene that is unable to express itself is called the recessive gene. F1 hybrid is the first generation of plants or animals obtained from cross-mating distinctly different parent types. When two individuals of the first filial generation are self-fertilized or self-pollinated, this is called the F2 hybrid. Next, self-fertilization is the fertilization of male and female gametes from the same individual. Only one individual will be involved there. So the monohybrid cross is the study of inheritance of one pair of contrasting characters. So you can see that like tall and dwarf plants. The study of inheritance two pairs of contrasting characters is known as dihybrid cross. For example, see the like round yellow seed and wrinkled green seeds. They are different. So to determine an individual is homozygous, dominant or heterozygous, the back cross or test cross is been carried out. A back cross is a cross between the F1 generation progeny and any of its parents. Whereas the test cross, it is a cross between the F1 generation progeny with the homozygous recessive parent. It is opposite to the back cross. So thus genetics will explain how the traits are passed from one generation to the another. And the principles governing to genetics were discovered by the Gregor John Mendel, and he is the father of genetics. So these terminologies are very important, children. Genetics, different genetics, alleles, phenotype, genotype, dominant, homozygous condition, heterozygous condition, and F1 generation. F1 generation is nothing but the filial generation, and it will be the first generation. Then, so phenotype means external, outer look, how it, the external morphological appearance is the phenotype, out. Genotype, the word gene, so it is a genetic makeup. Internal it is. Phenotype means external, genotype means internal. Then dominant means, you know, right, you might have heard the term dominant character. So one which is having the more character, so that character will be expressed, so that is why dominant. And homozygous and heterozygous, I hope you have understood that. That is homozygous means when a character is controlled by two identical, means they will be same, homo. Hetero means two different alleles, they are different. So that's what. So don't get this phenotype, genotype word, never get confused, and this homozygous and heterozygous also never get confused by these terms. So for today terminologies is enough. So next class you'll be studying about the laws, like law of dominance. In the same way, the monohybrid cross, dihybrid cross. So you'll be studying in your next class. So you have to remember this terminologies properly. Only then the whole chapter will be understood. Otherwise, you'll get confused. Okay, thank you.